Because the nucleus isn't the only difference. Hi biology friends, it's Sarah, your other biology professor, and it's time for Prokaryotes versus Eukaryotes DNA Replication Edition. Now, that's a lot of material to cover, so to keep it manageable, we'll focus on these areas. Let's start with the DNA itself. Prokaryotic genomes are much smaller than eukaryotic genomes and are typically organized into a single circular chromosome. The DNA loops onto itself in an area of the cytoplasm called the nucleoid. So that's where DNA replication happens, in the cytoplasm. Many prokaryotes will have additional bonus pieces of DNA, also often circular, called plasmids. Plasmids replicate independently of the chromosomal DNA using different mechanisms, so we're going to focus on the chromosomal DNA replication here. Meanwhile, the much larger eukaryotic genomes are organized into linear chromosomes, often with more than one copy of each chromosome. Humans have 2 copies of each. We're diploid. Eukaryotic DNA is packaged with proteins called histones in order to make it condensed enough to fit into the nucleus, the site of eukaryotic DNA replication. Now, you will also find DNA in mitochondria and in chloroplasts in our green friends, but again, different mechanisms involved, so we're going to focus on replication of the chromosomal DNA. To initiate DNA replication, proteins, including helicases, bind at specific sites called origins of replication. Proteins will separate the DNA strands so that they can be accessed by the replication machinery. Prokaryotic chromosomes typically only have one origin of replication, though you'll find some archaea with a few. Meanwhile, eukaryotic chromosomes have many origins of replication, hundreds to thousands. That's a lot of bubbles. Okay, so when are we all replicating our DNA? Well, bacteria replicate their DNA pretty much constantly. Once a cell has grown enough and accumulated a large enough stash of ATP and proteins, DNA replication is initiated. In fantastic growth conditions, bacteria are actually dividing faster than the DNA can replicate, and so a new round of DNA replication kicks off before the last one is even done. In eukaryotes, DNA replication is tied to the cell cycle, a highly controlled process. DNA replication happens during the S phase of interphase, while a cell is preparing for division. Specific S phase proteins are activated and recruit the proteins needed to initiate DNA replication. Cell cycle checkpoints ensure that DNA replication is complete and has occurred only once before allowing the cell to move to the next stage of the cell cycle. After initiation comes elongation, time to build that DNA. DNA replication is semi-conservative, so each strand of an original DNA molecule is used as a template to build a complementary strand. So our 2 DNA molecules produced in DNA replication will consist of 1 original strand and 1 newly created strand. Both prokaryotes and eukaryotes require a primer to build off of. Because DNA polymerases, our DNA building enzymes, can't start a new strand from scratch. In prokaryotes, primers are built by primases, special RNA polymerases that lay down a short RNA primer. Primers in eukaryotes are a little bit different. They're built by a complex of primase and a DNA polymerase, polymerase alpha. Primase starts the primer, laying down RNA nucleotides, but DNA polymerase alpha takes over and adds some DNA to the mix. It's possible that these extended eukaryotic primers provide a bit more stability for the working replication enzymes. Other DNA polymerases will build off of these primers. These polymerases can only add to the 3' end of a growing DNA strand, which means that each replication fork will have a leading strand synthesized continuously and a lagging strand synthesized in shorter Okazaki fragments. DNA polymerase III elongates both the leading and lagging strand in prokaryotes, while eukaryotes split the duties. DNA polymerase epsilon tackles the leading strand, while DNA polymerase delta handles things on the lagging strand. In eukaryotes, additional proteins will remove the primers, and DNA polymerase delta will fill in the gap left behind with DNA. While in prokaryotes, DNA polymerase I takes care of everything itself, removing the RNA primers while simultaneously filling that gap with DNA. Bacterial DNA polymerases fly through those replication forks at about 1,000 nucleotides per second, with archaea just a little bit slower. Turns out having less DNA packaging to deal with helps a lot. Combine that with the already much smaller genomes, and these guys can wrap up DNA replication in less than an hour. Compare that to about 50 nucleotides per second in eukaryotes. But because of our many origins of replication, we do all right. Mammals can knock out DNA replication in about 8 hours, even with our much larger genomes. Now, us eukaryotes are slowed down quite a bit by our much shorter Okazaki fragments, about 1/10 as long as the ones that prokaryotes make. Why? Well, our DNA packaging probably influences this as well. And shorter fragments means more fragments, which means more ligation, which is a slow process. When replication forks meet, the DNA replication machinery disassembles and the daughter DNA molecules are separated. In prokaryotes, you've just created 2 separate circular chromosomes. But in eukaryotes, the result is 1 replicated chromosome made up of 2 sister chromatids, identical molecules of DNA wrapped up with proteins that will later be separated during cell division. Those eukaryotic chromosomes may have shortened a bit as well. Because of our linear chromosomal structure, we have trouble filling in the gap where the final primer was removed from the lagging strand. So we add a bunch of repetitive DNA to the ends of our chromosomes called telomeres, so that we don't lose necessary genes when DNA replicates. Some cell types are capable of replicating the telomeres to keep them long, but most of our cells can't do that, so telomeres shorten as we age. Not a problem for prokaryotes with circular chromosomes, no ends. With the complexities of DNA replication, there are of course more differences and similarities between prokaryotic and eukaryotic DNA replication. If you know one that we've missed, drop it in the comments. Meanwhile, give me a thumbs up if you learned something about prokaryotes and eukaryotes and how they replicate their DNA. Subscribe for more biology content from me. Check out my website for tutoring resources, and I will see you next time. Bye.